There are 3 steps of translation. Initiation is the first step. DNA helicase splits the DNA. Half of the DNA finds a complementary mRNA strand. No. This out. Stop.
Keep going.
A new tRNA amino acid enters the ribosome at the next codon after the start codon. If its anticodon matches the mRNA codon, it base pairs and the ribosome can link the 2 amino acids together. The ribosome then moves forward and a new tRNA amino acid can enter the ribosome and the process is repeated. Termination. When the ribosome reaches one of 3 stop codons, for example, UGA, there are no corresponding tRNAs to that sequence. Instead, termination proteins can bind to the ribosome and stimulate the release of the protein, and the ribosome disconnects from the mRNA. When the ribosome is released from the mRNA, its large and small subunit disconnect. The small subunit can now be loaded with the new tRNA and methionine and start translation again.
There are 6 steps of transcription. Each set of 3. Step 1. Each set of 3 bases on the mRNA is called a codon. Each codon calls for a specific amino acid to be brought to the building site. This first codon is always AUG. It's called the start codon because it starts all the amino acid work and whatnot. Step 2. In the cytoplasm are molecules of tRNA. At one end of the tRNA molecule is the anticodon, a set of 3 bases that will match a certain codon. The other end of the tRNA can pick up and hold amino acid. Each tRNA can hold only 1 amino acid. Step 3. A tRNA with an anticodon that matches whenever a codon is placed on the ribosome clicks into place. Like, if those 3 went on a ribosome, which we'll use as this computer. The computer is a ribosome. Look at that's an amino acid. The codon and anticodon fit together to be like a jigsaw puzzle. Since the first codon is AUG, the first anticodon must have the matching bases UAC. And the U stands for uracil, A stands for adenine, G and C stands for cytosine. And you can't forget the G, guanine. Yes, G, guanine. Alright, step 3. The process moves along to the next codon, to the next codon, to the next codon, to the next codon. Let's say that the next codon is UCC. Then a tRNA with a anticodon of AGG will fit, and that will make an amino acid. Step 6. Steps 4 and 5 repeat until finally coming to the end of the mRNA that is the stop codon. At this point, the amino acid chain breaks off, folds up, and the protein is ready to use.